My name is Neil Foster. I lead Work Package 7, which is involved in molecular biology on the PROHEALTH project. Ivan has already mentioned that we've worked with the probiotic bacteria. At Nottingham, what we've been involved with is the development of diagnostic markers for production diseases. Something that I'm going to be talking about today are the development of diagnostic markers for two diseases in chickens. One is necrotic enteritis and the other one is coccidiosis. At Nottingham, what we actually did was we used a whole genomic array. So we actually looked at 44,000 genes expressed by chickens when they were infected with either Clostridium perfringens, which causes necrotic enteritis, or Eimeria species, which causes coccidiosis. From that, what we found was a bank of 11 genes which were downregulated by birds which had necrotic enteritis. We then tested these experimentally against birds which were infected with Eimeria maxima, which causes coccidiosis, and we found that they were differentially expressed but in the opposite direction. Now these were both with one strain of chicken, the Ross 308, which is a fast-growing broiler bird. When we tested this in Ranger Classic, which is a slow-growing broiler bird, we found differential expression, but only in one gene. So it seems in this case that we've developed a biomarker which, at least at the moment, looks like it can actually differentiate between Clostridium perfringens infections, necrotic enteritis, and Eimeria maxima infection causing coccidiosis. We've also gone on to look at the microRNAs, and we've looked at this in broilers which are clinically infected and subclinically infected, and we are actually using PCR now to actually validate the deep sequencing that we did, because in the deep sequencing we showed that there was differentiation between infected and subclinically infected. And so now we're validating this with PCR, and the results of that will come out within probably the next few weeks.